Hello everyone and welcome back to this lecture where we're continuing our discussion of Chapter 2 from your textbook. In the last part we talked about experimental evidence pointing to DNA being the molecule carrying genetic information. In this part we talk about the chemical structure of nucleic acids, DNA and RNA. DNA and RNA are made up of nucleotides. In DNA these are deoxyribonucleotides, and in RNA these are ribonucleotides. Nucleotides are made up of three main components: a nitrogenous base, a pentose sugar, and at least one phosphate group. An example nucleotide, deoxythymidine monophosphate, or DTMP, is shown here on this slide. The base here is thymine, the sugar is deoxyribose, and it has one phosphate group. It's worthwhile to note that a nucleoside consists of a nitrogenous base and a pentose sugar, whereas the addition of at least one phosphate group to a nucleoside makes up a nucleotide. One of 5 bases are typically found in each of the nucleotides that make up DNA or RNA. These are adenine, guanine, cytosine, uracil, or thymine. Adenine and guanine are structurally related to their parent molecule purine, whereas cytosine, thymine, and uracil resemble their parent molecule pyrimidine. So adenine and guanine are called purines, and cytosine, uracil, and thymine are called pyrimidines. In DNA, we find adenine, guanine, cytosine, and thymine, whereas in RNA, we find adenine, guanine, cytosine, and uracil. Pentoses, which are 5-carbon sugars, form part of the backbone of nucleic acids. Ribose is found in RNA, whereas 2-deoxyribose is found in DNA. It's worthwhile noting the numbering of the positions corresponding to each carbon in the pentose. You will notice that 2-deoxyribose is missing a hydroxyl group at the 2 position compared to ribose. Here I'm going to make a brief side note about notation for chemical structures. Your textbook uses 2 shorthand notations for chemical structures. Shown on the top left is the standard organic chemistry shorthand, with the points where lines representing bonds connect being understood to represent carbon atoms and the hydrogen atoms that are attached to them. The textbook also sometimes uses an alternative notation to draw sugars, which is shown in the bottom left of this figure. In this alternative notation, a straight line with a free end signifies a CH bond with the hydrogen atom at the end. Personally, I don't particularly like this alternative shorthand, and in my own notes and drawings, I'll usually draw sugars with the standard organic chemistry shorthand, where C atoms and H atoms attached to them are implicit. As I'd mentioned earlier, a nucleoside is a base plus a sugar. For RNA, you find ribonucleosides consisting of adenine, guanine, cytosine, or uracil attached to a ribose. Whereas for DNA, you find deoxyribonucleosides consisting of adenine, guanine, cytosine, or thymine connected to 2-deoxyribose. Once again, nucleotides are nucleoside phosphates, that is, nucleosides plus 1 or more phosphate groups. So they are composed of a base, a sugar, and 1, 2, or 3 phosphate groups. Each phosphate group is designated as alpha, beta, or gamma as indicated. It's also worthwhile to note that nucleotides store chemical energy. For example, ATP is the main energy carrier in cells. The hydrolysis of ATP into ADP and inorganic phosphate releases 30.5 kilojoules per mole, whereas the hydrolysis of ATP into AMP and pyrophosphate releases 45.6 kilojoules per mole. Overall, there is more energy stored in nucleoside triphosphates compared to nucleoside diphosphates, which have more energy than nucleoside monophosphates. The nucleotide units of DNA or RNA are linked together by phosphodiester bonds. Nucleic acids have a defined polarity. Each nucleotide is linked at its 5' end to the 3' end of the preceding nucleotide by a phosphodiester bond. A nucleic acid therefore has a clearly defined 5' end and 3' end, as in the example trinucleotide shown in this slide here, which has a phosphate group at its 5' end and a free hydroxyl group at its 3' end. DNA polymerization occurs with the addition of nucleotides to the 3' end of the growing strand. The 3' hydroxyl group attacks the incoming deoxyribonucleoside triphosphate at the alpha phosphate to form a phosphodiester bond with the added nucleotide, releasing pyrophosphate in the process. This is catalyzed by the enzyme DNA polymerase. The energy stored in the incoming DNTP, the deoxyribonucleoside triphosphate, drives the reaction. This energy is released with the cleavage of the pyrophosphate, or the diphosphate. The addition of nucleotides continues in this fashion. It is very, very important to remember that DNA polymerization occurs from the 5' end to the 3' end. DNA polymerase always synthesizes DNA from 5' to 3', and each nucleotide is added to the 3' hydroxyl of the growing strand. Another brief aside on notation. Sometimes your textbook uses this notation to represent nucleotides and nucleic acids. The pentose sugar is represented as the vertical line with the base attached to the 1' position and either phosphate or hydroxyl groups attached to 5' or 3' positions. I do not use this notation much myself, but you may find it in figures in the text. We'll move on now from talking about the chemical structure of DNA to discussing the higher order physical structure of DNA. This was solved in large part by the work of James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins in 1953. The key piece of evidence was uncovered by Rosalind Franklin, who found that bundles of DNA stretched out as a uniform fiber diffract X-rays, producing an interpretable pattern. The pattern shown in the image on the left may not mean very much to you unless you're an X-ray crystallographer, but Rosalind Franklin, who was an X-ray crystallographer, recognized that this simple pattern meant that DNA must assume a simple repeating structure. Based on this and other clues, Watson and Crick proposed a double helix model of DNA. For this, they shared the 1962 Nobel Prize with Maurice Wilkins, who had worked with Rosalind Franklin. At this time, though, Franklin had already passed away and could not receive the prize. However, she is widely recognized for her role in solving the structure of DNA. The other major clue for the double helix model came from Erwin Chargaff, who found evidence for base pairing in DNA. Chargaff analyzed and determined the base composition of DNA from various organisms and came up with a set of rules that we call Chargaff's rules. He found that for any given organism, the content of A's is equal to the content of T's, and the content of C's is equal to the content of G's. He found that the content of purines, that is A's and G's, is always equal to the content of pyrimidines, that is C's and T's. These observations were the first clues suggesting base pairing, A pairing with T and G pairing with C. Based on this, Watson and Crick proposed a mechanism of base pairing that involved hydrogen bonding, as part of their model for a double-stranded DNA double helix. In Watson-Crick base pairing, GC pairs form 3 hydrogen bonds and AT pairs form 2 hydrogen bonds. We know that DNA exists as a double helix, but depending on the conditions, this helix can twist in more than one way. Shown in this figure are A, B, and Z forms of DNA. In the conditions of the cell, DNA is found in the B form. The B form of DNA is a right-handed helix, with a little more than 10 residues per turn. It has a major groove that is 22 angstroms wide and a minor groove that is 12 angstroms wide. You can imagine a right-handed helix as a spiral staircase that you ascend by walking counterclockwise and that you can descend by walking clockwise. Here are several different representations of double-stranded DNA. In panel A, we see a representation that is stretched out and flattened, ignoring the helical nature of double-stranded DNA, but emphasizing the base pairing and the direction of the strands. The arrows here indicate that the complementary strands are oriented in opposite directions. You can see that the polarity going from 5' to 3' is flipped in the two strands. It is very important to remember that the two complementary strands of double-stranded DNA are anti-parallel. You can see the cartoon representation in panel B shows the helical nature of double-stranded DNA like a twisted ladder. The sugar phosphate backbones of the opposing strands are represented by the red and blue ribbons. We can see that Compared to the flattened and stretched out representation in panel A, when twisted up, either side of each base pair faces out from either a major or minor groove. The width of each groove is the distance between backbones of opposing strands. We can see that on one side of the double-stranded DNA, this distance is greater in the major groove than it is on the other side in the minor groove. The third representation on this slide shows double-stranded DNA in a space-filling model. The space-filling model provides a clearer picture when viewed in 3 dimensions. Here you can see more clearly the major and minor groove. The base pairing mechanism and double-stranded nature of DNA enables DNA replication. When they published their model, Watson and Crick made a famous understatement. They wrote, "It has not escaped our notice that the specific base pairing we have proposed immediately suggests a possible copying mechanism for genetic material." When it is unwound, single-stranded DNA is able to serve as a template for the synthesis of a new strand with a complementary sequence. Shown in this figure is an illustration of semi-conservative replication, which we will discuss in the next lecture. Each separated strand of DNA serves as a template for a new daughter strand. We will get into this in more detail in the next lecture, but for now we'll move on to discuss more of the physical properties of DNA. The base composition of DNA in different organisms can vary considerably. Even though we know from Caspars rules that the content of G's is equal to the content of C's and the content of A's is equal to the content of T's, the GC content, that is, the percentage of G plus C, can differ quite a bit between organisms. It can be as low as 22% or as high as 73%. These differences are also reflected in differences in the physical properties of the DNA. When DNA is heated, the non-covalent forces from hydrogen bonding holding the 2 complementary DNA strands together weaken and the bonds break. When the hydrogen bonds break, the 2 strands come apart in what is called denaturation or melting of double-stranded DNA. The critical temperature at which half of the DNA strands are denatured is called the melting temperature, or the TM. DNA with a higher GC content is observed to have a higher melting temperature. This can be attributed to the fact that GC base pairs form stronger interactions with 3 hydrogen bonds compared to AT base pairs, which only have 2 hydrogen bonds. This means that more energy is required to break these interactions, requiring higher temperatures. In addition to heat, DNA can be denatured by organic solvents or high pH. DNA also denatures more readily in low salt concentration. This can be attributed to the fact that in low salt, the repulsive force of the negatively charged DNA has greater influence. After 2 DNA strands separate, under the appropriate conditions, the strands can come back together. This process is called annealing or renaturation. For example, heat denatured DNA can be renatured by cooling. DNA that has been denatured in alkaline solution can be renatured by neutralization of the solution. And DNA denatured by organic solvent can be renatured by removal or dilution of the solvent. Some key factors influence renaturation of DNA. Obviously, the temperature should be below the melting temperature, but it should be high enough to avoid non-specific binding of DNA strands. Higher DNA concentration increases the likelihood that complementary strands will find each other, and so do longer renaturation times. When 2 nucleic acid strands from different sources base pair and form a double-stranded complex, this is called hybridization. Examples of hybridization might include a complex formed from 1 strand of DNA and 1 strand of RNA, or you might have 2 DNA strands from different sources that are complementary or nearly complementary. This might result from denaturation of 2 different double-stranded DNAs, which then reanneal to different partners. We can talk about DNA size in a couple of different ways. DNA can be measured by the number of base pairs, the molecular weight, or the length. Experimentally, we can determine the size of DNA by different techniques, such as by electron micrograph or gel electrophoresis. In addition to size, DNA comes in many different shapes. It can be circular or linear. Phage DNA is typically circular, as is the DNA of many bacteria. Eukaryotes, like us, tend to have linear DNA. DNA can also organize into higher order superstructures, like supercoiled DNA. You can imagine supercoiled DNA coiling up and wrapping around itself like a twisted rubber band, or perhaps in a more dated analogy, like the coils of a wired telephone landline. The size of DNA in the genomes of different organisms varies widely. The C value is the amount of DNA found within a haploid nucleus, or one complete copy of an organism's genome. You might imagine that more complex organisms have more DNA in their genomes, or a higher C value, but this is often not the case. What's known as the C value paradox describes the observation that the size of an organism's genome often bears no relationship to the complexity of the organism. Examples can be seen in this table from your textbook. Human genomes contain 3.2 billion base pairs. Some species of amoeba have more than 100 times as much DNA in their genomes, despite being much less complex. That concludes our lecture on Chapter 2. You can find additional reading in the textbook and exercises in the problem set and textbook problems.